Only physicians and qualified scientists working on the physician would be permitted to collect and use the tissue samples, but for this virtual lab, we'll put on our virtual doctor's caps. Our patient has arrived. Let's get started. We will collect a healthy tissue sample and a cancer tissue sample from our patient. We will need a scalpel and 2 empty samples to do this. We can extract the RNA from both tissue samples by dissolving them in a mixture of various organic solvents. We can separate the DNA proteins and as well as cellular components, leaving only RNA in solution. Next, we will mix our tissue samples on the vortex. As the vortex mixes the sample, the tissue will dissolve and the RNA will be released. Place the tube containing the healthy tissue sample onto the vortex. Now place the tube containing the cancer tissue sample onto the vortex. The samples have been placed into the centrifuge. The centrifuge will spin the sample and separate the RNA from the rest of the cell contents. Click on the lid to close the centrifuge. Press the green button to start the centrifugation. Then take the sample from each of the tube, the supernatant from each of the tube, and put them into the free jars. The sample contains several different types of RNAs: messenger RNA or mRNA, transfer RNA or tRNA, ribosomal RNA or rRNA. mRNA molecules but not tRNA or rRNA always end in a sequence of adenines known as the poly A tail. Only the mRNA reflects gene expression, so we will save the mRNA and get rid of other type of RNAs. We'll wash our RNA samples over columns filled with small beads that will only bind to RNA standard strands that have only poly A tail. The other molecules will wash away. Our columns now contain strands of mRNA attached to the poly T beads by their poly A tails. Now we will wash a buffer solution over the columns to detach the mRNA from the beads. Next, we'll make a DNA copy of the RNA. As we do that, we'll give the DNA some color. We'll add some labeling mix to our two RNA samples. We'll use green labeling mix for the RNA from healthy cells. For the cancerous cells, we'll use a labeling mix containing a red fluorescent dye. The labeling mix contains poly-T primers which bind to the poly-A tail on the mRNA, usually referred by the scientific community as oligo-DT primer. Reverse transcriptase, which is an enzyme which will synthesize a strand of complementary DNA or cDNA, and labeled nucleotides which will be incorporated into the new cDNA molecule. The nucleotide in this tube have a green fluorescent molecule attached to them. Reverse transcriptase has assembled the loaded nucleotide into a cDNA molecule, and the mRNA molecule has been degraded. Good work. Our tubes no longer contain RNA but now contain complementary DNA or cDNA. The healthy cell cDNA molecules are green and the cancerous cell cDNA molecules are red. This is what we will apply to our microarray. Two complementary DNA strands from different sources can also pair with one another. We call this process hybridization. For the next step is our experiment. We will take advantage of a key property of DNA, complementary base pairing. If two complementary DNA strands are mixed together, they will sooner find and base pair with each other, reforming a double-stranded DNA molecule. It doesn't matter where they will come from. They will do that even if they were not originally paired. Scientists call this hybridization, and it is the key to how DNA microarray works. Stuck to our microarray are little piece of single-stranded DNA molecules. A single spot contains many identical copies of same gene, and each spot on the microarray represents a different gene. On our computer database lists which gene is controlled in each spot. Now let us add the single-stranded cDNA we prepared from the healthy and cancerous cells to our microarray. Remember that all the cDNA molecules in our tubes represent human genes, and for every molecule of cDNA there is a matching spot of single-stranded DNA on the microarray. Pipette the healthy cDNA sample onto the microarray. let us take a closer look at what's happening on the surface of the slide. Most of the cDNA molecules have hybridized to their complementary DNA strands on the microarray. A few stray cDNA molecules did not hybridize. Now we will wash off the extra cDNA that didn't bind to the slide. Now let's analyze our data. To do that, we need a microarray scanner. First, we'll scan the microarray to see where the green labeled cDNA from the healthy cells bound. To simplify things, we'll explain just a section of the microarray. Many of the spots are green, but not every spot. The dark spots are green that were not transcribed in the healthy cells. Remember, not every genes get expressed in every cell type. Now we'll scan the microarray to see where the red cDNA molecules bound. Lot of spot again, just as for the green genes, but the red pattern looks different. It's clear that skin cancer cells and healthy skin cells have different gene expression patterns. But how can we start to plot this data and make sense of it? The microarray scanner can merge the red image with the green image and display the composite picture. Here are the red and green spots merged together. Any spot that continues both red and green cDNA shows up as yellow. A yellow spot contains a gene that hybridized to both green and red cDNA, which means that this gene was expressed both in cancer cell and a healthy cell. Genes that shows up a yellow spot probably aren't very interesting to us, since their activity doesn't change when the cell becomes cancerous. But look at all the red spots and the green spots. Red spots show genes that produces more mRNA in the cancer cells than in the healthy cells. They're the turned up in the cancer. Green spots show genes where expressions is turned down in the cancerous cell. You are a researcher interested in studying the genes that have an increased level of activity in skin cancer cells. What color spot would you choose to study? That's right, red spot experiment genes that are turned up in the cancerous cell.